All right, let's go. So the quiz for today, right? We had these two questions. We had the first one about the 5 most important macronutrients that cycle through ecosystems, right? Which one was this one? Yeah, it was A. And the class did pretty well on that. About 86% of you got that one correct. And on the second one, how about this?
3. I love 3.
You looked? Yeah, it is number 3. It is that species diversity and genetic diversity tend to be very high in natural systems, but they tend to be much lower in agroecosystems, primarily because we're reducing the total number of organisms that are in the system, since it's ideally just the crop, and we're also reducing the total amount of genetic diversity in that crop. We want the crop to be as genetically similar to itself as possible, right? So, 2 quick announcements before we get going. 1, just a reminder, lab will meet in room 119 today. We're not going out to the alfalfa field. It's not ready for sweeping. And then 2, we are past the academic drop period. So I've been getting a lot of emails from students who are missing lectures, and that's great that you let me know. At the same time, though, you're not obligated. Part of the reason I record the lectures is so that if you have to miss lecture, you can catch up on them later. So if you're going to be missing a lecture, you don't need to send me anything or show any evidence of why you left. You just need to catch up. And but obviously, if you're missing lab or some sort of graded exercise like a test, I'm still going to need you to let me know so we can work something out. So, just a reminder of where we were. We were covering population dynamics. So we were talking about density when we left off. Just density being the number of individuals in a unit of area. So basically, how much of an organism do we have in a particular place? And we were talking about how ecological factors can affect density. So what are things in the environment that can push populations up, things that can push them down, and how those apply to agroecosystems. And we were just sort of getting into the fact that we can group these factors into 2 major camps. Those that are density dependent, meaning that these are factors that their intensity depends on how many organisms are in the population. So essentially, if the population is really dense, the factor is more powerful. It kills a larger proportion of the population than it does when they're at really low concentrations or low densities. So normally we basically say the larger the population, the stronger the factor's effect. However, it can be the opposite. There are some cases where the more dense the population gets, the weaker the effect is, and the effect gets stronger as the population sort of becomes less dense. And then we talked about density-independent factors, or those where the intensity is the same regardless of the density of the population. So large and small populations tend to be affected in the exact same way. And we went over density-independent factors through an example, which if you recall, we had a shrubby sort of plant that was growing on in a valley. And then there's a flood that occurs, washes through, and anything that's living in sort of the lowest part of the valley gets flooded out. It's not flood tolerant, and so they end up dying away. And so when we go and we look at how many organisms in each population died, while a much larger number of organisms died in the high-density population than in the low, we see that the proportion that died is about the same, about 3/4, because the number of organisms that die in a flood, especially for something like plants that can't move, isn't dependent on the flood. It's not dependent on how many of the organisms there were. It's dependent on how much high ground there was. And if the high ground is about the same on each side of the valley, then we expect that you'll see about the same death rate. So we talked about this, and we touched on the fact that in agroecosystems, a lot of our control tactics are density independent. So essentially, we expect them to kill about the same number of organisms, pests, no matter what we do. So things like pesticides, resistant cultivars, GMOs, tillage, all of those things are independent of the density of the population. So let's walk through an example of sort of the opposite of this. Not the opposite, but the other category. which would be density-dependent factors. So again, factors whose intensity does depend on the density of the individuals in the population, such that the severity increases as the population density increases. So let's look at these same populations again. And let's say something else comes through, a different ecological factor than a flood, one that's not quite so random. Something like a disease comes through. So we can get a good idea of how diseases affect different densities of populations. So let's say disease strikes in the high-density population first. And this individual with a little white cross in it has been infected with some sort of fungal pathogen. Fungal pathogen settles into the plant, spreads out its hypha, eventually hits a reproductive structure, and puts out a whole bunch of spores into the environment. And those spores just sort of naturally distribute out from the host, and in such a way that it might form sort of a ring of contagion around the host, such that anything that's within that ring gets infected. They develop the disease, become contagious themselves. Those rings spread out until eventually you get to a point where everybody who was close enough to get infected was infected, and they all die off. On the flip side, in a low population, an individual gets infected, has a contagious period, but if the population density is really low, there's a good chance that there's no other organisms near enough to that organism to actually get infected. So that one dies, and that's the end of the disease. So consequently, if we were to look at this again, we could see that, yes, again, a larger number of organisms in the high-density population died than in the low. But if we look at these percentages, we see that 3/4 of the high-density population died, whereas the low one, we only lost 1 out of 5. A much lower death rate. And it's purely because the more densely you pack the organisms, the more likely it is that a disease will spread. And this is generally the pattern we see in density-dependent factors, which is that things that work better, that ecological factors that have a greater impact on dense populations, tend to be biological processes. They tend to be things like pathogens and diseases, predators, parasites, competition, essentially anything that feeds on something else. The more organisms you put in the system, the easier it is for them to find new prey, the easier it is for their offspring to find things to feed on. So you're going to increase their efficiency the more prey you put into the system. As far as competition is concerned, I mean, that makes sense as well. If you have a really large population and limited resources, they're all going to compete with one another more frequently than if you have a low-density population where they're all spread out and it might be a lot easier for them to find the resources they need. So those would be, right. And so in agroecosystems, generally how we see this play out is with biological control, right? That if you have a really high number of pests, you tend to get larger numbers of natural enemies in your system. So biocontrol works harder when you have larger pest populations, but also at that time, you might have so many pests that it's too late for biocontrol to keep it at sort of an economic level. All right, so that's it for density. Let's talk about distribution. So distribution can be thought of in a lot of different ways. It's essentially how organisms are spread within their habitats, and probably the easiest way to think about distribution, and the way we usually talk about distribution, is how they are physically distributed in their habitat. So basically, where are the pests in a field physically located? And we like this in IPM because it gives us a good idea of where to expect pests. So where are they? Where are they thriving very best? And it helps us when we're monitoring, because if we know where they're most likely to show up, it makes it a lot easier to keep track of them. So within pest systems, we tend to see 3 major types of distributions. We have nearly uniform distributions. These would be, and I say in pests, this is really the reality of how most organisms are distributed everywhere. But near uniform would be the fact that organisms tend to space themselves out in such a way that they have basically a territory area where they don't interact with one another. We have a purely random distribution. So organisms just kind of end up in a certain space. There's no real rhyme or reason to why they stay where they do. But you'll notice that random distributions oftentimes don't look super random. They oftentimes look a little bit clumpy, or you've got areas that seem really empty. The reality is that random systems don't look random. We usually expect random to look uniform, but that's just not how statistics works. So oftentimes randomness looks super clumpy. And then we have finally clumped distributions, where organisms tend to stay in little clusters together within their habitat. So I already clicked through, but what do you think would be the most common one you would see in pests in agroecosystems?
Random.
So I'm hearing random.
Uniform, because they plant a lot of row crops, and so they're all going to be in the rows uniform.
Okay, so I'm hearing uniform for that. Any reason you thought random?
Because like right there, when you drive around in the fields, the mites will be along the roads.
Oh, okay.
And they'll have hot spots, like
okay.
randomly throughout the.
Sure. So you're saying in your observation, they tend to be sort of randomly distributed around the edges, at least in the case of mites. Okay.
Fungal pathogens are usually clumped.
Fungal pathogens are often clumped. Thought I saw a hand in the.
I see some aphids that have been clumped in different areas of the field as well.
Mm-hmm.
But when you start talking about things like the horned tomato worm,
Yeah.
I don't think that's, yeah, you're not going to, I don't think it's going to be so random or so clumped because, as I understand it, the butterfly flies around or the moth flies around, lays one egg and flies to another plant.
Mm-hmm.
Right? But if you have a whole bunch of them, and like if you have row crops like tomatoes, you're going to be in rows.
Right. These are all very excellent points. And you basically led me perfectly into what I was going to say, which is that the reality is that even though you might expect them to be random or nearly uniform, the reality is most pests have a clumped distribution. And part of this is just along the lines of how do we define pests? So keep in mind that while you may expect insects to be very random, we have to consider that a lot of pests are things like fungal pathogens, are nematodes, are things that can't really go very far in their own lifetime. And so what they tend to do is they tend to infect a particular plant, put their inoculum in the soil, infect any plants that are near that, and they slowly just sort of spread out in these clumps. And then, like Aldo said, you've got things like aphids, which form colonies, and for the most part can't fly, so they kind of infest a single plant and spread out slowly from there as well, sort of like a pathogen would. Or you've got things like mites, where mites are transported on with dust oftentimes, or attack plants that are sort of dusty and dried out, so you might get more of a random distribution. Interesting thing you see with pathogens is oftentimes the distribution is clumped, but it's clumped in such a way that it's streaky. So you have like most of the inoculum right here, but then it streaks out. And this is because someone went through and tilled the field, and they spread out all of that infected soil in a streak. So sometimes you'll look at fields, and you can tell which way they were tilling, because you'll have one long streak going this way, and then a few rows down, you'll have a long streak heading out the other way, because they just sort of went out to the end, turned around, dragged the next clump the opposite way. So if you look at fields that have infection problems with some pathogens, you'll see weird streaky patterns where the plants aren't doing so hot. It's just because of how the pests got spread out. On the flip side, like Daniel was saying, we do see in some insects, especially leps, we don't see this clump, but we see more of a random pattern because the adults essentially just fly out, lay an egg or two in one site, and then find another plant to lay their eggs on. So they'll just sort of dot them across the field. Though the typical distribution we see is more random and not so much that uniform thing, because the moths don't really adhere to the straightness of the rows. They're just looking for the best possible host. So that's physical distribution. And physical distribution can be really handy as a PCA, because if you know roughly the way that the pest distributes itself, it makes it a lot easier to scout for them. So if you're looking for aphids, you might be heading along and you hit an infested plant. It might be worth your time to scout around in that area for a little while just to see how large the clump is. Because the reality is if you're going through and you hit that clump and then you just go out and sample the rest of the field, you may just completely miss all the other plants that are infested because they're all just in that one area, as opposed to being spread out evenly as some people might expect. So another way we can talk about distribution is not just in the physical distribution, but in the actual age of the pests that we're looking at, which is easily enough named age distribution. So age distribution is really just the proportion of individuals in each age cohort in a population. And so we could think of this as we'll get to that. So one way you could think of that is sort of this in this histogram right here, where we have two different populations with different age distributions. So here on the x-axis, we have the percent of the population that's in a particular age class, and then we have the age classes in months here on the y. So essentially we have like, in the 2, that would be all organisms that are 2 months or younger. In the 4, that would be all individuals in the population that are from 4 to 2 months. You just keep climbing up the scale, and yeah, so you get like 20 to 18 at the top. And so these were some age classes made of voles out in a field. And so we saw two very different populations. One, where the vast majority of the population was very young. Almost all the organisms, 60% of them were less than 2 months old. Whereas we had another population here, B, that was very evenly distributed. Most of the organisms were young, less than about 6 months, but you had a fair number of much older voles. Now, why does this matter? Why do we care about age distributions? Sorry? Mating, right? So age distribution can tell us when to expect mating events. If we're doing something like, say we're dealing with leps in a field where we might need to do mating disruption, having an idea of when the male moths may emerge can give us a good idea of when we should actually put out our treatments.
If they're being successful or not, like A's, something's killing them off.
Right, exactly. It could give us an idea of how successful they are in the environment, maybe how successful something we applied was. Maybe there's not too many older organisms because we applied a treatment a while back and we knocked that generation out.
It's a good indicator of how the population, you can expect the population growth where, I mean, in the case of B,
yeah,
compared to A, it's, okay, well, what are we going to do? We have this many. I think what I'm trying to say is that if, in the case of B, over long term, we can predict the population growth.
right.
Where in the case of A, we don't have that much data,
Mm-hmm.
right? We only have, what, 8 months of data collection? And then
Well,
there seems to be a population explosion. I don't know how many numbers there are, but
Yeah.
certainly to go from 0 to...
Right. We know that it's at least a relatively young population, that it's only recently shown up. So, exactly. So if I hear that, you're saying we can basically extrapolate population trends moving forward, especially in sort of this older population. And yeah, that's essentially where I was going with that, was this idea that we can look at the distribution of the organisms and we can get an idea of whether we expect the populations to grow, to stay about the same size, or to actually decline in size, based on how these distributions go about. And the general trend, and this isn't always 100%, but the general trend is that populations that are overwhelmingly young are poised to expand. You've got lots of young organisms that are basically sitting there gathering resources, and they're all going to hit, reproductive age right at the same time. So you're going to expect a lot of mating, a lot of eggs, a lot of offspring, a lot of seeds, all to basically show up at the same time. So that population is going to go from being, relatively limited to getting much bigger very quickly, which can be especially problematic if you're dealing with something like insects, where it's that young life stage that you're most concerned about, causing the most damage. On the flip side, stable populations look a lot like B does. They tend to be fairly evenly distributed. They'll have a little bit more in the young area, just because you see more mortality at young ages, so they tend to invest more in young. And because the older you are, the more likely you are to have died at some point. Well, actually, the older you are, the less likely you are to have died. By being alive, you are guaranteed to have not died at some point, just thinking statistically. But the older you are, the more likely it is that you will die. So anyway, stable populations tend to be sort of distributed more like this. So like Daniel said, we can basically have a better idea of what to expect from them. They're going to more or less stay about the same. And then declining populations tend to be the opposite of B. They tend to be, or A. They tend to have very large numbers of older individuals that are maybe past their reproductive stage, and we expect them to die off and to not leave behind a whole bunch of offspring for us to deal with. So, yeah. And so one final thing to keep in mind is that a particular population doesn't just have one way that it can go. It's a population that is growing will not just always grow. It will, as the environment changes, as the ecological factors change, we expect populations to shift from growing to stable to declining, or maybe to shift back to expanding. It all just depends on the time of year and the conditions. So these sorts of estimates can give us a snapshot moving forward, but they don't necessarily determine long-term what's going to happen. So where age distribution can be especially useful is in cropping systems where the pest is highly dependent on the particular crop they feed on, essentially where they have very few hosts that they can use, in which case you essentially see the pest life cycle synchronizing up with the crop, and to the extent that when we look at the pests, we see sort of discrete individual generations as the season progresses. We call this the cohort effect, the idea that organisms have distinct generational cohorts. So a good example of this would be something like a codling moth. It's a pest of apples and pears, and it's completely dependent on these particular plants. If you remove them from the system, codling moths don't do so well. And so essentially, they need to be at their prime, feeding and reproducing during the time of season when it's most efficient for them to do that. And so they sync up their life cycle very nicely with the trees. So what we've got here in this figure is the individual months as alternating bars, and then we've got different life stages of the codling moth. We've got these long caterpillars, we've got the individual adult moths, and we have the little dots that are eggs. And so what we see in the codling moth and in a lot of leps is that they tend to overwinter in one particular stage. In this case, they are larvae. So at the beginning of the season, we expect to see just a whole bunch of larvae that are overwintering, waiting for it to warm up so that they can come out and feed. And then once the environmental conditions are just right for them to transition to the next stage to start reproducing, we'll see a major transition. They'll enter that first generation. Those overwintering larvae will disappear. They will come out as adults. Those adults will lay eggs, and those eggs will hatch into larvae. And basically, we'll know that whenever we go out, that we can basically subscribe these to a particular generation, right? We can say that these are overwintering adults, these are first generation larvae and eggs, and we see this pattern just sort of repeat itself as the growing season goes through. We can distinctly tell a first generation from a second, from a third, from a fourth, based on the time of year and the life stage the organism is in. And this can become quite pronounced the longer the life cycles are, because you'll notice that as we get into these later life cycles, we start to see a lot of overlap, that there's like September where you've got potentially 2 different generations of eggs, caterpillars, and adults all at the same time. So it might be difficult to tell which one is which just by going out and observing. But where this is important is by knowing approximately where these life cycles sync up with the crop, and in timing, we can at least know that when the adults emerge, we can estimate approximately how long it is until we expect larvae to be coming out that might be more susceptible to treatments. It might give us a good idea of when biocontrol will show up, all sorts of things like that. So again, it's all about timing. And then finally, where are we at? All right. So finally, we can also look at age distribution in terms of not just how many organisms are alive, but how many of them are dead, or the ones that we expect to be dead. And we do this using something called a survivorship curve. So essentially, a survivorship curve has age on the x-axis And then on the y, we've got the percentage of survivors on a logarithmic scale. Basically meaning that instead of just going up by a step like 10%, 1 to 10 to 20 to 30 to 40, we go up by factors of 10. So at the halfway mark up this y-axis, we have a 10%. At the very top, we have 100%. So 90% of the population is crammed into this little half of a y-axis. And so in basically living organisms, we see three major types of survivorship curves. Basically, curves that tell us what sort of life spans we expect organisms to have. First is type 1, which is where the vast majority of the population survives through its young and through its middling years, and then you see a pretty hard crash once they hit a later life period. So a good example of a type 1 would be large mammals like humans, most domesticated animals, deer, elephants, things like that. Some birds are like this as well. Basically, mortality late in life. Type 2, which is the most uncommon type, but this would be mortality that's constant at all ages. So you're basically equally probable to die very young as you are very old. It just sort of stays the same the entire time. And you see this in some very rare cases of insects as well as some lizards, but again, not very common. And as far as pests are concerned and agroecosystems, we're really interested in most commonly seeing type 3. This is where essentially the vast majority of mortality occurs at the very beginning of life. So around the time eggs are laid and hatch, maybe the time that seeds go in the ground. So you essentially see here in this chart that just a little bit into the entire lifespan of the organism, we're already at that 10% mark, meaning that 90% of the organism died. So this tells us essentially two things about pests, which is that on one hand, pests are really susceptible at the young age. That's the time where if you're a weed, you're trying to find a piece of ground that you can actually survive in, and you need the ideal conditions to sprout. If you're a pathogen, you're looking for a nice moist piece of soil or a nice moist leaf to land on, which can be very random or hard to find. And also, this tells us that many pests expect to die in large numbers. And so their life cycles are set up so that they produce lots of offspring that they can afford to lose. So that a weed doesn't just produce 2 seeds, it produces 2,000 seeds. A fungus doesn't release a dozen spores, it releases 12,000 spores. They expect most of them to die. So even if you are out there scouting and you see a very small number of pests, that population can explode very quickly if the conditions are just right for their offspring. So we talked about this a little bit earlier. Population growth, what are things that can change population growth? And so I won't belabor these points, but just to go over them again before we move on into another section, remember that population growth is largely determined by internal factors such as birth and death, essentially with populations growing as birth rate goes up and decreasing as mortality rate goes up. But also there are external factors, things that are related to dispersal, how individuals move in and out of populations, such that as immigration or movement into populations increases, populations go up, and as individuals leave through emigration, populations go down, which led us to that delightfully simple equation I had last Tuesday, where basically populations go up if there's a high birth rate and if you have lots of immigration, and they go down if there's high mortality or high emigration. So something that ecologists really like is math. Ecologists like to basically take natural systems and they try to discover mathematical formulas that allow them to predict what will happen in those systems. And so, unsurprisingly, people have come up with mathematical models and equations that describe how populations grow and shrink. And we can apply these to agricultural settings, and especially in terms of pests. So with pest populations, we see generally 2 major ways that pests grow. The first of which is exponential growth, which is perhaps unsurprising, right? Exponential growth is basically just a growth pattern where growth starts off slow, and it increases by incremental steps until it starts growing very rapidly. And essentially just a simple model where the population always doubles itself over a certain period of time. The first time you double, you go from 1 to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32. Next thing you know, you're increasing by steps of hundreds or potentially thousands. So by the time you get to your 10th or 13th step out, your population is growing out of control. And this is the kind of growth we see in a lot of seasonal insect populations. Things like aphids that basically show up, clone themselves, and grow very, very quickly. And then you have large populations right around the time that you actually do the harvest. So the nice thing about exponential growth is we have a relatively simple equation to describe it. In this equation, what we have is the change in population size, or dn. So I guess I should start here. There is the population size, which is n. Generally, anytime you see an uppercase n by itself in ecology, that means population size. You have the rate of population change, r, and then you have the change in population, dn, or delta n, and delta t, or the change in time. And so essentially what it says is the change in a population size over time is equal to the rate of population change times the population size, which seems fairly straightforward, right? You have a certain rate of increase, you multiply that by the population size, and the population grows at that particular rate. And so because we're multiplying, and we're multiplying by the size of the population, as that size of the population gets bigger, the rate increases that not at a linear rate, but at that exponential, that we're just getting bigger and bigger and bigger by larger and larger steps. However, oftentimes, we don't see exponential growth because the reality is that nothing can grow exponentially forever. Eventually, you'll get to a point where the population is so large that eventually you'll run out of your food source. Or maybe a disease will strike and will wipe out a huge chunk of the population. Or maybe there will be so many of you that you're competing for the food sources that the population starts reducing itself. So essentially the idea is that in a lot of systems, there's some sort of environmental factor that stops the organism from growing exponentially forever. And we call that form of growth logistic growth, or log growth. It's also known as sigmoidal, or S-shaped curves. And this is the kind of growth we see in systems where there's a lot of density-dependent factors acting on the system. So if you have a lot of predation, a lot of pathogens, a lot of parasitoids, we see this sort of growth. And this kind of growth essentially is modeled by what looks like exponential growth at first. You have slow growth with a quick pickup as your population size gets large and that R kicks in. But then as you start to get towards this higher population level, this K point, we start to sort of slow down. Growth slows down, you start to teeter out until you hit that K point and you flatten out completely. The population hits sort of a steady state. And we call this K point the carrying capacity. Carrying capacity being the maximum population of a species that can be supported within an environment. And there's lots of things that can cause a K capacity or a carrying capacity to occur. It could be that predation I talked about. It could be the availability of shelter, the availability of food sources, of water. Essentially, if you've got something that is a limiting factor, it sort of causes this K effect, where the population has to flatten out at the top. So the nice thing about population growth in the sigmoidal curve is that there's an easy equation as well. You'll notice that the equation is essentially exactly the same as the one we had for exponential growth, but we've got a little modifier tagged onto the end, this little K minus N divided by K. And in this, N is our population size, and K is our carrying capacity. And so what you'll notice is that the way this works is essentially the closer that N gets to K, so the higher up this curve we climb, when K subtracts that N, this numerator is going to get closer and closer to 0. And so this entire modifier is going to get closer and closer to 0. Essentially what we're doing is we're calculating a percentage of how close the population is to the carrying capacity, such that if we had a population of, say, 100 organisms, n equals 100, and we had a carrying capacity of 1,000. Only 1,000 of that organism can live in that system. The way we would plug that into this equation is we would, within our little bracket, we'd have our k, or 1,000, minus 100, the n. We divide that by 1,000. So ultimately, if we calculated that out, that would be 900 divided by 1,000, which equals 0.9, or 90%. So essentially what we're doing is we're taking our normal growth, that r times n, and we're multiplying it by 90%. We're just cutting 10% of the growth right off the top. So that essentially, instead of growing, let's say the growth rate was 500. Instead of growing 500 new individuals, you only grow 450. You've cut off that top 10%. Similarly, if our n was much larger, say it was 900, we would expect this value to be 100 over 1,000, or a 0.1, which ultimately would translate to 10%. The population would only grow at 10% what was expected under an exponential model. So essentially, the closer n gets to k, the slower the growth is. Right, so n times r times n is reduced. And so yeah, this is a very common sort of growth. And the reality is that most all pests are held to this carrying capacity. But in agroecosystems, we oftentimes don't let them hang out in the ecosystem long enough to get to that point, right? Because if economic damage starts down here, we're not going to wait for them to hit that carrying capacity. We're going to go out and spray them. Alternatively, we might harvest the crop before they ever get to that really high carrying capacity level. And so, for us, oftentimes what pest population growth will look like is this, but it's because the carrying capacity is way up here. It's somewhere where we're never going to get because we're going to take care of the problem before that. All right. And I believe after this we go to community. So this is the last thing within populations. And this is, I just wanted to talk about the idea of ecotypes. So ecotypes, basically what we're doing is we're recognizing the fact that not all populations behave the same. We may have populations of the same pests, but located in very different locations. And that those two populations may look very different from one another in their features. And it's because natural selection will basically act on individuals in their own particular environment to allow them to adapt to that environment. And if we have two very different environments from one another, we may get two very different adapted populations. And we would call these different populations ecotypes. Or in other words, locally adapted populations of a species that allows it to live under specific conditions that may have previously been detrimental. So essentially, a pest adapted to local conditions. So where this matters is that oftentimes a pest or any organism can't expand out of the area it's living in because it doesn't have the right genetic tools. It doesn't have the right adaptations to spread into a new habitat. But over time, they may, under certain pressures, expand into new areas. In agriculture, a major concern we have is pathogenicity. The idea that not all bacteria, fungus, or viruses have the ability to infect crops, but if we plant crops all the time and are always exposing these bacteria and fungi to these crops, they may over time gain adaptations that allow them to move into those crops. So we'll have emergent diseases that we didn't have before. And so it's potentially the same species of bacteria infecting the plant as it was before, but it has adapted itself. It's become a new ecotype. Alternatively, pests can adapt themselves to also endure other factors such as climate, so that maybe at some point you had a particular fungus that didn't move into the Imperial Valley because there just wasn't enough water there, but if they develop some drought tolerance, they can move in, and resistance is a big factor as well, that organisms can adapt themselves to overcome particular pesticides. This happened with the potato psyllid. So potato psylla didn't used to occur in California. They had populations up in sort of the Pacific Northwest, Idaho area. You had a couple randomly distributed populations in the central United States. And a big thing was they couldn't really get into California because it was hard for them to migrate over the mountains. It was hard for them to come up through sort of the more desert regions of California and Mexico. But ultimately what they did develop was they developed a lot of pesticide resistance, so that when they eventually were introduced accidentally to California, the normal pesticide regimens that we would use to control them that would just sort of incidentally kill them no longer applied. And so they could just sort of move up into Southern California and feed there. All right, so do I want to get into communities? Yeah, I've got 8 minutes. Let's go into communities. I've only got one slide. So, we've gone through individuals, populations, now we're in communities. And if you recall, communities are the level of organization in which all populations interact with one another. So this would be the level at which plants, animals, and microorganisms share the same habitat. They interact directly or indirectly with one another. Excuse me. And that's basically the long and short of it. And this is clearly important in agroecosystems, because this is the level at which pests would interact with plants, biocontrol would interact with pests, all of those sorts of things. But we're going to touch on those in more detail as we move into the different types of control. We're going to talk about plant-insect interactions a little bit later when we talk about things like resistant crops. We'll talk about biocontrol in the biocontrol section. And so we'll touch on aspects of community a little bit later. So the only real thing I wanted to touch on was, firstly, the idea that sometimes in communities there are organisms that are more important in terms of interactions than others are. And we call these guys keystone species. These are a small number of species that basically have a disproportionate influence on the entire community that they are in. And so by removing this species or manipulating it, you might actually change the entire ecosystem very substantially. In agroecosystems, keystone species can be relatively easy to identify, as they oftentimes are the crop. So clearly, if you get rid of the crop, you're going to change the whole cohort of organisms that you see organizing around it. But you also see this in natural ecosystems. In the Pacific Northwest, the sea otter in some areas was hunted to extirpation, so they no longer existed in that area. And what they found was that by removing the sea otters, you no longer had any control on starfish, and the starfish basically increased in numbers, wiped out the native mollusk populations. No? I'm seeing head shaking. Oh, thank you, sea urchins. You're right. It was the sea urchins.
Sea urchins, without the control, that ate all the kelp beds.
That's it. See, I'm not a native Pacific guy, but yes, thank you. I was kind of pulling that off the top of my head. But yeah, so sea urchins ate the kelp beds, and once you remove the kelp beds, that's kind of the whole center of the ecosystem. And so, all right, 10 points to Dan. There you go. All right, well, there we go. Hold on. 10 plus 10. Aren't you going to be disappointed when you find out 10 what? All right. Yeah. And then finally, just the idea, just something to keep in mind is the idea that community interactions are very complex. And so oftentimes, when you think you're pulling on, John Muir said that if you pull on one string of nature, you find that everything is interconnected. And it's very much the same in agroecosystems, that you might be trying to control one organism and unintentionally cause all sorts of manipulations in areas you weren't expecting it to occur. All right. So, ecosystems. I think with this one, we are fully one lecture behind. So, let's just stop there. That'll leave me a nice place to pick up next time. And then we can basically just call it a loss, say we're one lecture behind, and